Thank you very much. I've had an absolutely splendid day at MedGenome today. Splendid. And I thank you for having me here. I'll talk of looking for needles in haystacks, which is of course quite easy. All you need is a magnet. It's very straightforward. And before I get to the needles in a haystack, let me start with showing you this slide, which I hope you can see clearly. It's a slide that goes to the very root of all cancer immunology. It shows patients who are immune suppressed because they have received kidney transplants. So in order for them to continue functioning with another person's kidney, and for the immune system not to reject the kidney, they have to be immune suppressed. These patients are at a high risk of getting cancers. They are at 25 times higher risk of getting non-melanoma skin cancer, 10 to 15 times higher risk for various cancers of thyroid, head and neck, gynecological cancers, and some lymphomas, about 2 to 10-fold higher risk of a bunch of other cancers, kidney, bladder, colorectal, and so on and so forth. This is a solid clinical observation made more than 20 years ago, that if you are immune suppressed, you are at a high risk for getting cancer. This tells you very clearly that the immune system has a role to play in genesis and progression of cancer. You can also do this in the laboratory. You can take mice, healthy, happy mice, and inject them with a tumor, and the tumor will grow, and at some point will kill the animal. Mouse, rat, hamster, guinea pig. On the other hand, if you immunize the animal with tumors that cannot kill because they have been irradiated, for example, actually tumors either won't grow at all, or will grow and regress, or the growth will be considerably slowed down. Just like with smallpox and polio and measles and mumps and rubella and the tens of vaccines that we give ourselves today, you can immunize against cancers just as easily. It's a remarkable phenomenon because cancer is, by and large, not an infection. It's not measles, it's not a virus, it's not mumps, it's not rubella, and yet you can immunize against cancers. That tells you that cancers must contain something that is not there in the normal host, because you can't immunize yourself against your liver or your spleen because that's your own body. The fact that you can immunize against cancers means tumors must have something that is tumor specific. We still don't know what that thing is. That's the needle in the haystack. But the needle is there because you can immunize. Very basic biological understanding. There's a problem in this phenomenon. The problem is there's a paper from a while ago. George Klein, the first author in this paper, died last month at the age of 91. George made this observation that you can immunize mice against tumors, but there's something else. And here I'm using the schematics. If you have a colony of mice, let's say 5 mice, and you inject a tumor in each mouse, so you have 5 different tumors with the same carcinogen. All tumors are the same type histologically. And you have now tumors A, B, C, D, E, and F. If you immunize mice against tumor A, they're immune to tumor A, but not to B, C, D, E, and F. If you immunize mice to tumor D, they're immune to tumor D, but not to other tumors, even though all these tumors are of the same type, induced by the same carcinogen in a colony of mice that are completely inbred, and yet they are individually distinct. Every time you have a tumor, it's a new tumor. Every time you have a tumor, it's like a tumor that has never existed before. This phenomenon was so strange that people tried to test this in many ways. A group at Weizmann Institute in Israel took one mouse and injected this with the same carcinogen on two different flanks, got two tumors, and tested and found they were both distinct. Same animal, same tissue type, same carcinogen, two tumors independent, as if they don't know each other. Someone extended this to 42 by 42 matrix. Someone at Fox Chase Institute in Philly in 1970s, they were all distinct. Every time you have a tumor, it's a new tumor. And this was sort of ignored as some kind of peculiarity of animal tumors. Of course, you couldn't test this in humans because you can't. Fortunately, you can't go on injecting people with carcinogens, even though you may want to, but you generally can't do that. So, struggling with this problem, as to how could you have so many antigens, so to speak? Every time you have a tumor, how can you have a new set of antigens? About 25 years ago, I published an idea that is in this grainy old picture, but I have replotted this in a new shiny slide, and that is shown here, the shiny slide. So I argued that, as we know, each time a cell divides and goes from one cell to two cells, the replication of DNA is pretty good. It's pretty faithful, but not completely faithful. Each time a cell divides, bacterial cell, mammalian cell, human cell, any cell, there is an error in replication, net of repair. Even after all the repairs that have happened, the errors can be few or can be many, but every time a cell divides, any cell divide, normal cell, cancer cell, any cell, there are errors. I argued at the time that because these errors, they are just random errors, hence every time there will be some new errors. And I argued in that paper that therefore every tumor will have, of course, the driver mutations like oncogenes like RAS P53, but it will also have a number of incidental mutations that will be completely random. And because they are random, some of them would be seen by the T cells as foreign. And I think you all understand the T cell recognition of antigens, so I would not really go into this at all. But I argued that you're starting with two normal cells that are completely identical, same transforming mutation. Then you have random passenger mutations, and they're presented onto the MHC molecule, and you end up with tumors that are antigenically unique. That this uniqueness comes not because there are millions of tumor antigens, but because the process of generation of mutations is random, and because it works on a large base of 6 billion base pairs in a diploid human genome, and so you will have a new pattern each time. That was the idea. At the time I suggested this, there is no way to actually test this idea. In 1980, when I was a fellow at Sloan Kettering, I used to sequence 100 base pairs a week, and I was a champion. I was a champion. Nobody sequenced faster than me. And so the possibility that I would ever test this idea was never really there. It was just a pipe dream. So, of course, times have changed. We can sequence more than 100 base pairs a week. So what we can do now is what happens all the time at every place. It happens right here, across the hall, yes. You can take a mouse or a patient, you can take their normal exome sequence, you can take the tumor exome sequence, you can find the mutations, and you can try and predict the neoepitopes. So the essential point that I'm going to talk about is a negative point, that we don't really know how to do this. We don't really know how to tell the ones that are good from all the ones that are useless. We all have various ideas on them. Amit has some ideas, he showed you, I have some ideas, but we don't really know them. The Genentech group has some ideas, as do various people, but no one quite knows. No matter how much we claim that we can, but we can't. And this is what I'll talk about in the rest of my talk. This is the needle in the haystack. It's there, but how do you tell which is the one that can be used to protect against cancer? So, by and large, this is what we do. We say, can this be processed by the proteasome? Now we all understand that all proteins get chopped up and pieces of peptides get presented on the class 1 molecules. The affinity with MHC, are they actually presented by MHC class 1? And how is the binding of the mutant peptide to the wild type peptide? These are the most common factors that everybody, not almost everybody, but everybody considers in their pipeline. And the rest of the talk, almost all of the rest of the talk, I shall devote to discussing some of the issues. This is a small enough group that I framed the rest of it more as a discussion than as a formal talk. So I'll talk about rules to define neoepitopes, rules to find the needles. I'll talk about value of mice. I'll talk about T-cell response in mice and humans. I'll talk about something I know not next to nothing, but almost nothing about, mass spectroscopy. But you have the next speaker to talk about that, and she knows much about it. I'll talk about adjuvants, and I'll talk about the TCR recognition, about which no one knows almost anything except in this building. So these are the subjects I'll cover. So a couple of years ago, we published a paper where we took many of those epitopes that we had identified as being mutated, and we tested them for their ability to mediate protection from tumor growth. So you take a peptide, you immunize mice with it, and then you challenge all the mice with tumor cells, and you wait and see who gets the tumor and who lives, who dies. And for example, here each line shows a tumor growing in a single animal, and you can see mice immunized with this peptide. All of them got the tumors. With this peptide though, 4 out of 5 mice did not develop a tumor, or tumor grew and regressed. In this particular peptide, every single mouse was protected from the tumor. This was the first such demonstration at the time. Now, I won't go into detail. So these are the 1, 2, 3, 4, 5 or 6 peptides we showed that showed complete protection from tumor growth. The higher this bar, the bigger the tumor size, and so these had no tumors at all. The problem is that if you look at these peptides, which are shown here in red, and you look at their affinity for binding to MHC class 1, the affinity for this one is undetectable, meaning it is less than IC50 of 70,000 nanomolar. If you look at this one, undetectable, 51,640 nanomolar IC50. Undetectable, undetectably low affinity. These peptides had such low affinities that normally you wouldn't even screen them. They have such low affinities, you would say, they don't mind, they're garbage. They're the ones that in Amit's slide, before they had one of his slides, they were way below the 500 nanomolar mark. These peptides you would normally not even consider. And so actually at that time, I thought that maybe this was not an immunological phenomenon. Because if you can't bind to the MHC, how can the T cell look at it? So it must be something weird happening. And so we tested if it is immunological or not. And not to take you through this in any detail, but just to show you that these are peptides showing tumor rejection. If you deplete CD8 cells from them, the tumors grow quite nicely. So the response is CD8 dependent. Here you have MHC class I binding peptides, or seemingly non-binding, that are generating CD8 response and causing tumor regression. So this idea that there is some threshold of affinity is completely, my goodness, even I cannot see this, and that's a good thing too. Wow. Well, just to make the point that this is another set of peptides that we have since characterized, and we're in the process of now publishing, where we demonstrate that the peptides with very, very poor affinities that you can detect in a different tumor still mediate tumor rejection that is CD8 dependent. So let's talk of these rules a little bit here. I've said a bunch of papers here, and all to make the following point. I mean, obviously, the peptide has to bind to the MHC if it's generating CD8 dependent response. It's not possible that it doesn't. So my thinking is that the problem is how we measure the affinity. And how do we measure the affinity? We take a recombinant MHC class I molecule, we take a peptide that binds to it, we bind it in vitro, we compete this in vitro with a bunch of peptides in different quantities, and by this competition, we determine the affinity. Now, let's just take a moment to think about how does the MHC peptide interaction happens in vivo. The peptide is chopped up in the steps that Amit had pointed out in his talk, chopped up, goes through the tap, and there is some selection there, gets into the ER lumen. There are a whole bunch of chaperones, like Tepessin and others, and calreticulin. They come and help it bind to the class I molecule, and then it's transported onto the surface. Nothing at all in comparison to what we are doing in vitro. The two phenomena, the way we measured it in vitro and in vivo, are completely unrelated. So I think the problem is not that these peptides are low affinity, it's just that the way we measure them in vitro measures something that's completely irrelevant in vivo. That's my belief. I found a paper here that just came out a couple of months ago from a mass spec group whom I'm sure you would know, where actually they showed that although most peptides that they eluted were high affinity, but a very significant proportion of peptides were much below the 500 nanomolar affinity range. A paper was presented at a meeting recently by Bob Schreiber showing the same thing. I've listed here things that I would not go into detail, but there has been another formula from Model von Kettering by Tim Chan's group suggesting that there's a tetrapeptide pattern that can predict if it's a good peptide or a bad peptide. This idea is completely debunked in a recent paper that just came out a month ago from the first author of that paper. They just simply misanalyzed the data as they themselves clarified in a correction of their own paper. This third algorithm is the one that we use where we suggested that it's not the affinity that's important by itself, but the difference in the affinity between the wild type and the mutant. So it's the differential affinity that's important. I won't go into any detail here, but there are various ways to look at the rules, and some of them are completely useless, and some of them have some utility. So here is a paper that appeared about a year ago, more than a year ago, where they took 110 patients with melanoma. They got a checkpoint blockade, a CTLA-4 blockade, ipilimumab, and they looked for response data, patients who did well or didn't do well clinically. And it's a very nice paper because they provided in the supplement information in great detail about total exome sequencing, RNA-seq data of every epitope, every patient, really in great detail. They concluded that overall mutational load, neoantigen load, etc., were associated with clinical benefit. However, no recurrent neoantigen peptide sequences predicted respond to a patient population. That there is no way to tell these. Well, So here is the analysis that we did with that data. This is the data of 110 patients. Each patient has between 10 and several hundred neoepitopes. So this is a vast amount of data. And so this is a cartoon that suggests what we did. So let's say there are 3 patients, patient 1, patient 2, patient 3. And we looked for correlation of survival with the affinity of peptides as traditionally measured. We also looked for correlation of clinical activity with the score that we developed, which we call the DAI, that is differential binding with the wild type and the mutant. And because each patient has many epitopes, just to give you an example, so this patient 1 has 5 epitopes. It becomes a number constant here to show the point. So these 5 are in a pretty narrow range. These are in a broader range. These are in the broadest range. The highest DAI score is here or here or here. I'm not sure if you can see this table, but basically what it shows is that when you look for correlation of clinical activity with the peptide affinity, there is no correlation, neither the mean, nor the max, nor the min, nor the range. Nothing correlates with clinical activity. If you look at the DAI, the differential affinity of the wild type and the mutant, the highest, so there's no correlation with the mean DAI, but with the max DAI. These values here, there is a significant correlation, and in the range of DAIs, there's significant correlation. To further analyze this, and this is all unpublished, and we've shown that starting from the top, the correlation remains at the top DAI value, second, third, fourth, fifth, and then it begins to be lost. This is being submitted. Similar data have now been generated by a group at UPenn, where they have characterized every patient in the TCGA database for whom exome sequence information is available and transcriptomes are available and clinical data are available. And they demonstrate that our formula of looking at differential affinity is the only one that correlates with clinical survival. That's being submitted for publication at the same time as our paper right now. Just to make the point that we really don't know much, we know very little, and of course I have told you my take on these. How do we ever actually tell? How will we ever find out what the rules are? So we have done a very painful analysis. And I mentioned to some of you when I met you today. We have taken 2 tumors and we have decided we will make peptides corresponding to every single mutation. So this comes to about 328 in one tumor and 658 in the other. So we have made peptides that are 10 mer on this side, 10 mer on that side. So you have every possible epitope covered. Basically 21 mer peptides, about 1,000 peptides. We're immunizing mice with these 1,000 peptides and testing mice for immune response and for tumor rejection. With no preconceived notion, none at all. We're not using any algorithm. If there is a mutation, we'll just make a peptide corresponding to 10 amino acids this side, 10 that side. We'll immunize mice, look for T cell response, and look for tumor rejection. These experiments are painful and should get done at some point. This is just what I just told you, what we are doing. And actually, we have some early data. Interestingly, so the higher the bar here, the more the anti-tumor activity. Some peptides seem to elicit very strong activity, and some peptides actually suppress immunity. Completely unexpected. I didn't expect this to happen. We're going into this in more detail to find out what's happening. But this was done. This is the largest project anywhere in the world of testing peptides with no preconceived notions to test for activity, not just for CD8 response, but for their ability to mediate rejection of actual tumors. So we will know what's happening here. I want to talk about mice for a moment. And I just want to make some basic facts that are perhaps known to this group, but are often not as known. People say a lot that it's very easy to cure tumors in mice. I've heard a lot of very intelligent, educated, informed people say that. When someone says that, I urge you to ask them, have you ever cured a mouse of a tumor? And the answer is no. If they tell you they have, they're wrong. I'll tell you why. I want you to understand this. Tumor rejection in mice are very rare. In humans are equally rare. Everything that has failed in mice has failed in humans. Everything that has worked in cancer therapy in mice has worked in humans. So what happens? Typically, people take a healthy mouse, 6-week-old, or an 8-week-old mouse, inject it with a peptide on day 0. Day 7, they inject it with another peptide, with the same peptide. On day 14, challenge these mice with tumor cells. And turns out the peptides work, the tumors don't grow, like I showed you before. And they say, well, this peptide worked. This is my pipeline, this worked, this is great. Then you go and take a 60-year-old man or a woman who has stage 4 or stage 3 or stage 2 tumor. And you give them peptides by the same method by which you cured the mice. And nothing happened, the patient dies. And you say, look, it worked in mice, it did not work in people. This is idiocy. This is stupidity. If you really want to compare the mice and human, take a 1-year-old mouse. That's like a 60-year-old person. Inject it with the tumor. Let the tumor grow to about this size. Now treat it. How many experiments of this sort have been published? 0. 0. So when people say that what works in mice doesn't work in people, they just don't think straight, they just don't know the literature, and they certainly haven't cured any mice. Anyone who has tried to cure a mouse knows how difficult it is. Checkpoint blockade worked quite nicely in mice, works in humans. There are not many examples of things that work in mice. So I just wanted to make this point clear. And also I say it very clearly, because saying clearly also allows people to disagree very clearly. So I'd love to hear any comments in disagreement with this, because then I will learn. But I certainly don't know of any example where people have actually cured mouse tumors and that hasn't worked in humans. Nobody has cured mouse tumors of the kind of tumors that we treat in humans. None. So this is quite problematic. So what do we love doing? And I was talking to some of you today about this earlier. We love measuring T-cell responses in people and in mice. And I'm saying something that's very difficult for me to say as an immunologist, that the T-cell response data have been quite useless. Now let me comment on this. They've been quite I mean, obviously, there is no question, I don't think anybody would doubt, or at least they may, but they shouldn't doubt, based on data, that T-cells play a huge role, maybe not the only role, but a huge role, almost certainly the central role in tumor regression. At the same time, there are examples galore, and I've listed some of them here, where T-cell response data have misled us. Take, for example, the MAGE antigens. Some of you old enough would remember MAGE antigens. MAGE antigens were the tumor antigens that would cure cancer. GlaxoSmithKline spent about $3 billion, 3 B as in boy, $billion, conducting over 10 phase 1, phase 2 trials, conducting the world's best, largest phase 3 trials ever done in cancer vaccines. They screened 8,000 patients with melanoma and lung cancer, immunized 2,000 patients of melanoma and lung cancer combined, a phase 3 trial that went on for 5 years, beautifully monitored, beautifully studied, showed no clinical activity. And everybody was in shock. My goodness, MAGE elicits great T-cell responses. Why did it fail? That's the point. That's the point, that the T-cell responses are essential, are necessary for tumor rejection. They're not sufficient. If you look for a vaccine that generates CD8 T-cell response, you will get a vaccine that generates CD8 T-cell response. You've got to be clear as to what you're looking for, because you will find what you seek. And of course, then one says, well, what else can I measure? And that's a good question, for which I don't have a good answer. All I say is that when you measure, know what you're measuring and know what you're not measuring. Actually, it's only when you know what you're not measuring, you'll begin to think about how to measure what you can't measure. If you assume that what I'm measuring is all there is, then that's all that you get. There are lots of data in mouse and human systems, and I'll spend a minute to talk about. Actually, so I'll point just to show you my age, as you can probably guess by my beard. But it is not that this is something that is obvious only in hindsight. It's not that, oh, what could you do? You couldn't tell. Now we know this. No, it was clear even at the time. 1996, I published this article saying, do human cancers express shared productive antigens? Or is the antigen stupid? And other lessons from vaccine therapy. It was clear even then, based on lots of data. But this is what happens when people lose track of what reality is. Then facts become alternative. Then you have wiretapping. I think, Amit, you didn't wiretap me, did you? Although I see a wire here. So let me just, to sort of drive this point home, let me talk of ovalbumin. We all know ovalbumin because it's such a great molecule. $42,000 Dalton chicken egg molecule that has this beautiful epitope that we all know, synfical. So let's just look at ovalbumin. So I told a graduate student to study ovalbumin, and he looked at me as if I was crazy or if I thought he was crazy. But I think we found some really wonderful things with ovalbumin. And I won't tell you all of it, but just some things. So if you transfect a tumor cell with ovalbumin, a chicken protein, this is a very highly immunogenic protein, generates great T-cell responses and antibodies. Would the tumor grow? The tumor shouldn't grow. It has this massively foreign antigen. The tumor grows beautifully and kills the animal. So then you say, what's this foreign antigen doing? You scratch your head. But it's an interesting thing to bear in mind. Secondly, if you look for CD8 response to synfical in these mice where this tumor is growing, these mice where this tumor is growing, vast amounts of this protein, and you say, let me look for immune response to this powerful epitope, synfical. There is no response. You won't find it. And yet, if you want to use synfical to immunize the animals, you can immunize with it quite nicely. And you say, wait a second, you're wrong every which way. Why does the tumor grow? Why doesn't it immunize? Why is there no immune response to it? And yet you can immunize against it using synfical. If you were trying to find an epitope for an overexpressing tumor, you would not find synfical because there is no T-cell response to it. And yet it's a potent vaccine against the tumor. Just to bear in mind that there are so many ways to get screwed here. We've got to know what we are measuring. And OVA is such a great example. So, skydiver. I think this part here, so I'm actually done with most of my, how am I doing for time, Amit? Do you have any idea? Do you have 5 minutes more? Yes. Okay. So, this kind of has nothing to do with the needles in the haystack issue. I'm done with talking about that. What I've said is that the haystack is there, the needles are there. We all have various ideas of how to find them, but no one quite knows how to go about finding them. Let me just switch here a little bit and talk about this patient. He has an actual name, but I call him a skydiver. That's the name I gave him. He's a boy, man, I don't know, what is he? He's a boy who presented with stage 4 melanoma at the age of 20. Advanced disease. He was treated at the NIH with TIL therapy, which didn't do anything for him. He came to Sloan Kettering, where he received a checkpoint blockade of CTLA-4 and PD-1. And he had 3 tumors, which you can perhaps see here, or maybe not. And 2 of these tumors kind of began to either stabilize or shrink, but not by much, and 1 tumor kept on growing. We checked to see if the patient had any other tumors anywhere, and PET scan was clean. He had no other tumors. Normally, stage 4 disease, you don't do surgery because surgery doesn't do anything. But we thought he's 22 at the time, 23. He has no other tumors, only these 3 lesions. Let's take them out and send him home. And most likely he will be dead anyway. But we could do this. So we took out his tumors. And we analyzed these tumors by exome sequencing, RNA-seq, and also his normal blood as a control for exome sequencing. It so happened that the patient survived. It's now been 4 years. He's now all of 26 years of age, and he's free of disease. We probably cured him by surgery. So why am I telling you this story? We predicted a large number of epitopes in this patient based on a number of criteria. We tested the T cells from this patient, as he is now, for immune response to all of these. Actually, we tested only against 24 peptides. We drew blood in December. And just to show you how that we know how to do these assays, you have positive negative controls. Here, basically looking at CD8 positive cells, CD3 positive, CD45 positive that make interferon gamma, and they make IL-4 and TNF. Peptides 2, 12, 26, and 29, they generated cells that made CD8 positive cells that were interferon gamma positive, or interferon positive, or both positive. So 4 and actually 1 more, perhaps 5 of these were positive. 5 of these 25 peptides were actually positive. Why did I show you all this? Oh, wait a second. I did miss something here, didn't I? You know what? I'm not done with my list. So why am I showing you this here? I'm showing you this for one reason. There's the point that I mentioned. One part of this whole equation of tumor immunity that nobody understands is the T cell receptor recognition. How does T cell receptor look at it? And we know that a single T cell receptor can interact with tens of thousands of different MHC peptide complexes. There is a huge amount of redundancy in this recognition, and nobody quite knows how to do anything about it. I think it's fair to say that nobody knows how to do anything about it, except maybe perhaps guys at MedGenome. I think as I have said to you, in my opinion, what you're doing will be a huge step in us being able to determine what the epitopes are. And I will just leave it at that. Let me come to something I know almost nothing about. So actually, our second speaker, Beatrix's PhD mentor, Don Hunt, was the person who introduced me to mass spec in 1993. And mass spec has been much in news about using to try to detect peptides. And I repeat, saying that I know nothing about it, but let me say a couple of things. There's an abundance bias, so much so that you can take tumors that express OVA and have syncytial that's immunologically active and fail to detect syncytial by mass spec in peptides eluted from MHC class 1. Some of the most highly protective peptides that we have shown are of very low abundance, very low abundance, which would have a lower likelihood of being picked up by mass spec. And I could be wrong here. You can speak up either now or in your talk, but I worry about this issue of abundance. We have learned about peptides being spliced. That peptides actually, it's just each time you think that you begin to gradually understand some major principle in biology, something happens and punches you in the face, and peptides getting spliced. Just can't even begin to think about this paper in Science a few months ago, low affinity peptides, and there's a paper highly acclaimed that came out recently, really sophisticated paper, and of course they talk about clinically relevant epitopes, but very serious caveats that I won't go into right now, but I can talk to you about this. Because I don't know mass spec well enough, I just don't know if it can help us in routine use as a high throughput mechanism. It's something that can tell us some rules to understand that we can use, but by itself as a high throughput tool, not quite sure about it. Adjuvants. So I have two adjuvants here, three adjuvants here. Adjuvant A, B, and C. We tested them on two neoepitopes. One is a really bad neoepitope, it does nothing, and the other is a good neoepitope. Adjuvant A used with a good neoepitope, good CD8 response. With adjuvant B, good CD8 response, and with adjuvant C, also good CD8 response. Not great, but good CD8 response. Tumor rejection. No tumor rejection with adjuvant A, some tumor rejection with adjuvant B, and outstanding tumor rejection with adjuvant C. Same neoepitope, but no tumor rejection with adjuvant A and outstanding with adjuvant C, where CD8 response is equivalent. The bad epitope, outstanding CD8 response to adjuvant A, poor with adjuvant C, of course, with no tumor rejection. There's a huge disconnect. Please. Meaning it causes tumor rejection. It's good. It doesn't cause it's bad. Yeah? No, it does not. No, it says negative, minus. Yeah, negative, negative, negative. Negative, positive, plus here. Got it? So even though it generates great CD8 response, beautiful CD8 response, but no tumor rejection. So just to make the point of the importance of adjuvants. I will skip the next 4 or 5 slides and just end with talking about the affinity, how we need to figure it out, the peripheral tolerance, how to predict antigen presentation, and understanding T-cell recognition. And that is where you guys, I think, are at the cutting edge. Let me end with 2 additional slides. So this is a paper in 2003 in New England Journal of Medicine about patients with ovarian cancer. Ovarian cancer is the most lethal cancer of women. It's diagnosed quite late because it has no specific symptoms, just bloating, discomfort in the belly. So most often when it is diagnosed, it's stage 3 or stage 4. 75% of patients diagnosed with stage 3 or stage 4. Almost all patients get surgery and chemo, and almost all the patients respond to surgery and chemo. They do very well. Many are seemingly cured of disease. Almost all the patients come back with disease, and almost all patients die of disease. So it's a lethal disease. It kills pretty quickly. And this paper and many others after that showed that if you have infiltrating T cells in the tumor, then the tumor progresses slower than if you had no infiltrating T cells. You live longer if you have infiltrating T cells than if you had no infiltrating T cells. Very, very clear, solid data in at least 3 or 4 large studies in different parts of the world. It's clearly an immunologically important tumor. So we're now doing a clinical study, phase 1 trial in ovarian cancer, where we do a lot of things, as you can tell. But very simply, we extract DNA and RNA from the tumor and normal tissue, and we predict the needles in the haystack by 2 different criteria. One of high affinity, the other of differential affinity, and we predict about 10 epitopes. We immunize patients with these epitopes, and we look for immune response. We also take a single cell suspension of these tumors and divide them into lymphocytes and non-lymphocytes, and we sequence 6,000 individual cells by total RNA-seq. 6,000 individual cells, and also 6,000 or somewhere near there lymphocytes, to get a sense of heterogeneity of the tumor population and the T cell activity. And basically, the endpoint is, of course, safety, feasibility. It's a phase 1 trial, but also immune response. And obviously, we'll monitor survival. We plan to ask permission to add another arm here, where we will combine this treatment with CTLA-4 blockade and go on from there. This trial just started. The IND was approved about a year ago. We applied for 3 years ago. It was approved a year ago, and the first patient just got accrued about a month ago. I think I'll stop here. I'll just mention some people. So all the bioinformatics work is done at School of Engineering, and Ion Mandoyu and Sahar Al-Sisi. Our X-ray crystallography work is done at Notre Dame by Brian Baker and his colleagues. We do some work with BMS. These are people in my lab. And actually, every single person here has contributed to various parts of the experiments. I think I'll stop here and take any questions. Thank you.